Thanks, Corinne, for that very kind introduction, and thank you all for coming along this morning. And I'd especially like to thank Jeff Bland, who's really changed the way I think about diet, its impact on health, and how it underpins many of the connections between our own microbes and the microbes around us. So this is going to set the stage for what you're going to hear the rest of the morning, telling you about the methodology that we use to understand the microbial world that's within us and that surrounds us. I have a few disclosures. None of them are particularly relevant to this, although I will mention day 2 very briefly in one slide. And the learning objectives, first off, I want you to learn about the approaches we developed for the Human Microbiome project that allow us to map microbes from birth to death and across the whole body. Second, I want you to be able to identify how human-associated microbial communities relate to those that we find around us in the environment. And then third, I want you to get an understanding of how integrating the chemical world with the microbial world can help us understand systems like the cystic fibrosis lung, but also the space around us, and ultimately how it will improve human health. So our world is very much a microbial world. All around us we see complex results of microbial activity. And amazingly, there are 100 million times as many bacteria on Earth as there are stars in the universe. And the vast majority of these bacteria are unknown at this point. And what this means is that today, microbiology is the ultimate in big data science. In fact, Julian Davies, who was on the advisory board for the Human Microbiome project, was quoted in Nature as saying, "Once the diversity of the microbial world is catalogued, it will make astronomy look like a pitiful science." So we're very much taking these numbers you might have thought of as astronomical numbers and reclaiming them as microbiological numbers instead. Microbes also outnumber us even in what we like to think of as our own bodies. So each of us has about 30 trillion human cells, but we have about 39 trillion microbial cells. And so by that measure, you could say that we're just 43% human. But you might be thinking, hey, wait a minute, it's the 21st century. Do we really care about counting cells, or should we think about this at the level of our DNA? And when we think about that, it becomes even more remarkable. So each of us has about 20,000 human genes, but astonishingly, the size of our microbial gene catalog, as revealed by the Human Microbiome project and other large-scale projects like this, somewhere like between 2 and 20 million microbial genes. And so by that measure, we're at best 1% human. And there's a tremendous amount of excitement at the moment about systems biology, systems medicine, systems nutrition, but it's hard to do systems anything if you're neglecting 99% of that system. And what's most shocking is that the genes that we're neglecting are the ones we can change. They're not fixed at conception, but we can change them during our lifetimes with lifestyle factors that we have control over, and we're increasingly understanding how. If you take something like obesity, for example, which I've studied for the last 15 years, it's true that human genes have a tremendous impact on obesity, but microbial genes have even more of an impact. And today, for example, I can tell you with 90% accuracy if you're lean or obese based solely on the microbes in your gut. So on the one hand, this is a cool trick from a technical perspective. On the other hand, we don't think it has a lot of potential as a test for obesity, because I bet you can tell which of these people is obese without doing any DNA sequencing at all. Right? But if you do that classification task as lean or obese, even if you look at every single nucleotide polymorphism in the human genome linked to obesity, you can only do that classification task, lean or obese, with about 57% accuracy based on human genes versus 90% accuracy from microbial genes. And this makes a lot of sense given about what we know about the timing of the obesity epidemic, where obesity took off dramatically from 1985 to 2010. And for that to be evolution in the sense of a change in human gene frequencies, either all of the obese individuals would have had to have had a tremendous number of children, or all of the lean individuals would have had to have had no children at all. And that would have been very noticeable had that been the truth. And one thing that's shocking about all these microbial genes is that we're losing them right now. So when we study people like the Hadza hunter-gatherers, who I had the pleasure of visiting to collect samples back in 2014, when we work with groups like the Hadza and the Yanomami and other people living very ancestral lifestyles, what we find is that they have completely different microbiomes from people living in modern society. And so this effect is so dramatic that when you just plot all the points, and I'll describe the technique that leads to these principal coordinates graphs later, but when you just plot all the points, the main axis is from least industrialized to most industrialized. And people like the Hadza, the Yanomami, people in Papua New Guinea and Central Africa and so forth, what we find is that they have entire phyla of microbes that we find in a lot of our close relative species, but you won't find in anyone who's taken up farming, let alone anyone who participates in an industrial lifestyle like ours and sits in air-conditioned rooms and so forth. And what instead we see is that this community is replaced by a much lower diversity community with completely new kinds of microbes coming in and colonizing our bodies. So it's almost like we took the rich inner rainforest of an ecosystem that's within us and on our bodies and replaced it with a cityscape where only the rats and the pigeons survive and not much else. And I'm sure you're all familiar with Rachel Carson's work from the 1960s, documenting how efforts to get rid of mostly single insect pests with compounds like DDT led to a biodiversity crisis throughout the ecosystem, notably the deaths of huge numbers of birds. And my colleague Marty Blazer at NYU has recently sounded a similar alarm call in the microbial world in his wonderful book, Missing Microbes, that goes into detail about all the ways that we're depleting our microbiomes, everything from overuse of C-sections and infant formula to things like overuse of antibiotics, lack of dietary fiber, and other ways that we're failing to maintain diverse microbiomes. And there's certainly strong indications that these consequences really matter, because if you look over the 20th century, as advances in public health and advances in medicine led to the conquest of one infectious disease after another caused by a single organism pathogen, ranging from measles to TB, at the same time we see an explosion in asthma and type 1 diabetes, in multiple sclerosis, in Crohn's disease, and in many other disorders. And what amazes me is that in 2002, when these graphs were published in the New England Journal, none of those diseases were known to be linked to the microbiome, whereas today we know all 4 of those and dozens of others are linked to the microbiome in the gut, and we can cause or cure them in animal models by modifying the animal microbiome, although therapeutic applications to humans are still a ways off. So I thought I'd tell you a bit about what tools we have to map our microbiomes and how we find all this stuff out. And back in 2007, it was published in 2008, my lab did an experiment where simultaneously we doubled the number of marker gene sequences about environmental samples that were in GenBank, and we simultaneously gained insight into hundreds of microbial communities from the human lung, from rivers, from the air, and from the Guerrero Negro microbial mat, the most diverse microbiome that we know of on Earth still. And what we were able to do was introduce a technique called barcoded pyrosequencing, where we amplified DNA from each of these samples using the polymerase chain reaction, introducing an error-correcting primer on each sample. And then what we could do is we could take hundreds of those PCR products, mix them together, and run them on one sequencing run of what was then called next-generation sequencing. But what was amazing about this is we collected 500,000 sequences. At the time, the Sanger facility down the hall from my lab still charged $8 per sequence. So if we'd done it with the old technology, it would have cost $4 million. We were able to do it for just $12,000 on a run that took overnight. And this is what enabled efforts like the Human Microbiome Project and the Earth Microbiome Project. Perhaps most amazingly, that same amount of DNA sequencing today would cost you just 75 cents. Right? So what's driving a lot of this technology is that what used to cost $4 million is now less than $1. And that's why you can apply it in all these different application areas. And that cost is continuing to decrease. Now we're looking at optimizing other parts of the pipeline like sample handling, data entry, and other stuff that costs more than actually collecting the sequences. But the problem is that we get huge piles of data that we have to interpret. And to illustrate this problem, if I just show you a whole bunch of numbers, it's very difficult to understand what's going on in this data set, right? No clue about what patterns might lurk in there. It doesn't help you if I organize them into their natural order. And you might think, well, what I need is more precision on each number. So if I give you that, it's also very unhelpful and there's no clear pattern, right? But if I take exactly the same data and I do a transformation on it visually, so in this case I turn each of those numbers into a shade of gray and arrange them in the same order, it's immediately obvious what's going on. And A lot of what we're doing at the moment is trying to take these inscrutable piles of numbers and sequences and counts of taxa and genes and give you similarly compelling pictures. But what's a problem at the moment is in the field there's a big focus on getting more and more precision out of one sample, so doing deeper sequencing, looking at longer reads, that kind of thing. And going back to astronomy, it's a little like studying the sky with your only tool being spectroscopy, where you're trying to get a more and more detailed spectrum, gazing at the same star over and over again in more depth. And that's fine, and it's a very useful technical exercise, but you're never going to understand the whole sky and the whole universe that way, where you need many pixels, in our case, many samples, and you need to stitch them all together into a pattern. So my lab's been focusing on that for the last few years. And to give you an idea of the problem, some of you have probably seen this type of OTU table, where for many samples, for many kinds of microbes, you have a count of each kind of microbe in each sample. And this is just 9 samples, and it's already impossible to look at, and it's just a small fraction of this table. And in a recent study we did with Peter Dorrestein here at UCSD, he's my close collaborator on mass spectrometry, we had not 9 samples, but 900 samples that we needed to turn into a picture. And so we turned it into this picture, which is immediately compelling. The LiDAR scan that we took, that we used as a reference frame for this spatially, just going to rotate that around in 3D, took us about 30 seconds to collect with a handheld unit strapped to an iPad. But then it took us hours to swab all the surfaces and all the people, and then months to do all the DNA sequencing in mass spec. And you can see this one microbe, so the scale on this is blue is the least of something, red is the most. You can see one microbe that lights Peter up very brightly, but not the other 3 of us. And what we're seeing is Synechococcus. If you know any microbiology, you might think, wait a minute, isn't that a marine microbe? And sure enough, it is. What you're seeing is that Peter swims in the sea more frequently than the rest of us, although he hadn't done it for 2 weeks before we did the sampling. And you can see this environmental trace incorporated into his microbiology and left behind. We can also do the same sort of thing for mass spectrometry. So this is showing you a piece of what's called a molecular network, where we take chemical data from hundreds of samples and look at it. And again, it's very difficult to understand the data in this sort of format. But once again, we were able to map the chemical data onto the same scan of Peter's office. And what I'm showing you here is one molecule that lights 3 of us up very brightly on our faces and hands, but you can see it doesn't touch Peter at all. Although, the molecule that we're looking at is caffeine, right? So this shows us a very obvious interpretation of why you might have that spatial distribution. And 3 of us drink a lot of coffee or tea, whereas Peter completely abstains from it, although you can see someone has spilled some on his floor at some point. And while this is undoubtedly the most expensive way to find this sort of thing out right now, you can imagine all kinds of applications for this in your offices, in the homes of your patients, and all kinds of settings to understand what is the connection between humans and the environments that we live in. And this is just one molecule of the 10,000 molecules that we measured during this experiment, many of which have similarly interesting spatial distributions. And we've been working with the National Institutes of Justice to develop this into a lifestyle profiling method. So the idea is that we can take chemical or microbiological traces that you leave behind on objects that you touch, such as your phone, or objects that might be at a crime scene, where we can tell a lot about you from these chemical and these microbiological traces in cases where, say, a fingerprint doesn't allow us to identify a person who's touched an object. And even if you do have a fingerprint, that doesn't really tell you anything about a person, the way things like traces of medication or traces of molecules derived from food or sunscreen or other lifestyle factors can give us insight into. So unfortunately, to a large extent, this is still not how microbiology is studied clinically. So if I ask you to name a microbe out of your gut, you're probably thinking of this one, E. coli. And it's true that we could isolate E. coli from pretty much everyone sitting in the room in a clinical lab. But the reason we know so much about it isn't because it's a dominant player in the gut ecosystem, because in a healthy adult, E. coli typically makes up between 1 in 1,000 and 1 in a million of the cells in the gut. But what it's great at, and why we know so much about it, is it's great at growing in a petri dish or in liquid culture. But the problem with studying microbes in this culture-dependent way is it's kind of like going down to the zoo and looking at each microbe in its individual cage, and knowing nothing about its environment or what it's supposed to interact with. And so to get around this, what we have to do is we have to study the microbes where they are in their natural environment. And to do this, we use a technique pioneered by Norm Pace called environmental sequencing, where what we do is we get samples from the human body or from soil or water or whatever, and we extract DNA from them. Then we amplify them by PCR. Then we sequence them, and then we used to blast them and match them up to what they were similar to in a database called GenBank. But ironically, this is getting less and less useful now as there's more and more environmental sequencing, because now you're not going to hit E. coli or some other organism you've heard of. You're going to hit some unidentified sequence from one of these studies. So instead, what we have to do is we have to slot the sequences into a phylogenetic tree and understand them based on their closest relatives. The problem with this approach of identifying microbes one at a time by their sequences in the environment, though, is it didn't really scale. And to illustrate this, I'm showing you a paper that was in Science, one of the best scientific journals, back in 2005, where you could get a paper in Science by sequencing a handful of new sequences, so those are the ones here in bold, then integrating them into what was already in the database. And then in this case, they had just 4 samples that they were clustering together, where you can see that 2 of those samples are more similar to each other than the other 2, but that's about all you can see. And the problem with this approach is that as we got more sequences, it was harder and harder to deal with. So for example, in this paper, they're trying to solve a very important environmental problem, which is how do termites take wood and turn it into fuel. And so they're addressing a very important problem, and I don't want to pick on them specifically, but you're reading through the paper and you come to figure 3, and it's one of these trees like the ones I just showed you, with some new sequences with short labels, and then what's already in the database with long labels. And that's okay, but then you come to figure 4, which is another tree, and figure 5, and figure 6, and a whole lot of additional figures with the tragic inevitable conclusion, right? And so our brains just aren't meant to handle data on that scale. And to get around the problem, we took a leaf quite literally from Darwin's book. So for those of you who haven't read his work on the origin of species or haven't reread it recently, it's often considered fairly dense, right? So the first edition was 502 pages and only 1 figure. So people who read it for the pictures are often disappointed. But I'd argue that you shouldn't be, because the 1 figure that he went to the effort of including was this first representation of the idea that the phylogenetic tree of grouping organisms by common descent and their shared characteristics was the right way to get a handle on life's complexity. So the next step in this was not to do it one at a time, but to do it for a whole community. And this was really pioneered by Kathy Lozapone, who was one of my first graduate students at Boulder. And I initially recruited Kathy for a completely different project to do a mechanistic study of a molecular machine called type 3 secretion that my lab was working on at the time, and that various pathogens, including salmonella, used to do nasty things to our body's own cells. And about a year into the project, Kathy told me, you know, Rob, I'm just not that into type 3 secretion. I'd much rather not be growing these bacteria in the lab, but I'd rather do something with evolution and sequences and trees. And I told her, well, Kathy, you know, I don't want to stop you from doing what you really care about, but you need to realize that type 3 secretion is a very hot area of science, whereas the microbial diversity stuff that you want to do, like what Norm Pace does, no one really, there's no funding for it, no one really cares about it. And even though he's a National Academy member and a MacArthur Genius Award winner, when he wants to work on microbial diversity, he has to move to a new university, get startup funds, spend it on this microbial diversity stuff until they're gone, and then he has to move again. And his lab actually runs on NIH grants about RNSP, which is another molecular enzyme. But Kathy persisted. She really wanted to work on this stuff. I think it's fair to say that she's done fine for herself. So she's now a professor at the University of Colorado at Denver with tenure. But even when she was an assistant professor, she was already one of the most highly cited people at that institution. And what she introduced that's had such a large impact, whereas in contrast, we never wound up publishing anything on type 3 secretion, despite working really hard on it for years. So it really exemplifies why it's important to enable your students to follow their passions, even if it's against your advice. But the technique that she introduced that had such a profound impact was this idea that we could exploit evolutions to compare entire communities at once. So the intuition here is that if you have 2 identical communities, like on the left, everything you find in the red community you'll find in the blue if you sample them completely. So none of the branches on the tree are unique to one of those communities. All of them are shared, and we define that distance as 0. Whereas in contrast, over on the right, you have two unrelated communities, so you can't move from the red to the blue and survive and evolve and add the molecular change that would lead to branch lengths on these phylogenetic trees, where the tips are modern sequences and the interiors are hypothetical common ancestors that we infer from the sequence similarity. And so all of the branches lead only to the red community or only to the blue community, never to both, and we define that distance as 1. So what's so great about this is that we can take Darwin's insight that there is a universal tree of life that connects all the microbes everywhere, all the organisms everywhere, and take pairs of communities and compare them pairwise on that tree. So here I'm showing you a tree with red, yellow, and blue, where we compare red to yellow, red to blue, and yellow to blue. And then we summarize those distances in a distance matrix, and then we can use techniques like principal coordinates analysis to get a spatial layout of how the samples are related to each other, or hierarchical clustering, or various other visualization techniques that have been developed by data scientists over the last 70 years. So with this technique in hand, Kathy initially decided she'd look at everything. And so she went to GenBank and put together a set of samples that literally spanned the whole planet, everywhere from the poles to the equator, extremes of temperature, pH of just about every biotic or abiotic factor that you could imagine that might influence a microbial community. And the question was, with all this complexity, was there even going to be anything that structured all those microbial communities relative to one another? And much to my surprise, the answer was yes. This very clear pattern emerged where the saline and the non-saline samples separated from one another very cleanly. You can see a few points in blue there. What those are estuaries, where you really have a mixture between the saline and the non-saline environments. And you're probably thinking, hey, wait, what about pH or temperature or other things that must matter more to the microbes than salinity? Well, it turns out that we're wrong about that in our intuitions. So if I show you high temperature environments from a hydrothermal vent at the bottom of the ocean or a hot spring in Yellowstone, those just look like the other saline and non-saline communities respectively. So temperature doesn't matter to the microbes nearly as much as salinity or about 10 other factors that are more important than temperature is. So this was very interesting, but then in additional work with Ruth Lay, now director at the MPI, and Jeff Gordon at WashU, we were able to find some environments that really were extreme from the microbial perspective compared to this data set from around the world. And when we add these microbes to the graph, you can see it squashes all of those samples, the saline-non-saline split, up into just one side of the graph, although that split is still present. And the new axis it introduces explains twice as much of the variation in the data as the saline-non-saline split. So you might wonder, how far do we have to go to find these extreme environments from a microbial perspective? Do we have to go to the top of a mountain or the bottom of an ocean or what? And amazingly, we don't have to go far at all because they're right there within us. What we're seeing is primarily the mammalian gut, which has a completely different microbiome from anything that we find in a free-living environment. And then what's in between, like those blue dots, what those are things like human skin, human oral samples, and other interfaces between ourselves and the environment that we inhabit. And another way of displaying these data is in what's called a bipartite graph, where what we do is we have two kinds of dots, where one kind of dot represents samples, and the other kind of dot represents kinds of microbes here at about the genus level. And we draw a colored line between them if a particular sample had a particular microbe in it, and that line is colored by the sample type. So here, blue is the human gut, green is the vertebrate gut, red is all those free-living environments, and then this little sprinkling of other colors between the big green and blue region and the red region, which shows all the things like the skin, the vagina, the mouth, and so forth. And so what's amazing about this is it shows a very profound way where microbiology connects the human body to the planet, where there's just a few kinds of microbes that are shared between us and our environment, and most of those are on the surface rather than in the interior. And we can also see how the human microbiome relates to the microbiomes of other mammals. I won't show you this today, but essentially we look very much like the other omnivorous primates, like the chimpanzees and the baboons and so on, although across different species of mammals, there's a profound influence of diet. So this really gives us a new way of seeing how we connect to the planet in terms of the microbes that we share and in terms of the microbes that our body is able to keep out. But there's another way, there's another very profound way that we're connected to the planet, and that's through food. And I was amazed by this paper that just came out in PNAS recently, where essentially what we're looking at, sorry this is a little bit cut off, But so this is from David Tillman's lab at the University of Minnesota. On the x-axis, we're seeing the relative risk of mortality of eating different foods. And then on the y-axis, we're seeing the averaged relative environmental impact. And you can see that there's a very strong positive correlation between those two things. So the foods that are bad for you are also bad for the environment and vice versa. Whereas in general, the foods that are good for you, like vegetables, fruits, potatoes, and so on, also have very low environmental impacts. And I think this is a very important message to get out there, that very frequently what's good for you and what's good for the planet are aligned with one another. So the success of these early studies relating microbes around the world were one of the factors that led to the Human Microbiome Project, and the technologies like UniFrac and barcoded sequencing that I showed you really enabled us to do the Human Microbiome Project, which was a large-scale effort funded by NIH. The largest single component was the Healthy Baseline study, where we looked at up to 18 sites on the body, which, as you can imagine, is a lot of places to stick a Q-tip. In the first paper, we reported on the first 242 people, and then in the second one, it was up to 300. And we collected 4.5 trillion bases of DNA, so 1,500 human genome equivalents, using a mixture of 16S ribosomal RNA profiling, like I showed you before, the PCR-based approach, and what's called shotgun metagenomics, where we just take all the DNA and fragment it and then try to puzzle it back into pieces and complete genomes and so on. And so the great thing about this is we had an unprecedented amount of DNA data concerning the human microbiome, but that was also the terrible thing about it. And to illustrate the scale of this problem, here I'm just showing you the first file of data from the HMP. And it's pretty hard to tell who lives where in the environment from this, right? Despite the fact that we're fundamentally doing ecology, you probably can't even tell that's an oral sample, let alone the sequence signatures, so let me determine that. And this is just the first 0.1% of this file, and then there's another 17,000 files just like it. So you can see that we really have our work cut out for us interpreting it. And this is a problem increasingly not just on the research front, but for you clinically, right? Because I bet a number of you have had the experience of a patient coming into your office, either with a report they got from American Gut, which is our citizen science project that I run here at UC San Diego, or from a company, where they come in with a huge grin on their face and they tell you, hey doc, I have this great news for you, which is I had my microbiome done, and now I have this list of 1,000 species they found in my gut. With all this data, you can tell me what's wrong with me, right? And what are you going to do? Refer them to your colleagues in psychiatry for being crazy enough to think you can do something with that? And a lot of what we're trying to do at the moment is to make it not crazy and to come up with visualizations and methods where you could take that profile and integrate it with other profiles and actually get something out of it. And so the processing pipeline called QIME stands for Quantitative Insights into Microbial Ecology. What it does is it encapsulates UniFrac and a lot of other processing methods and lets you take the data from a sequencing run and integrate it with other data sets. So when we apply this to the Human Microbiome Project data, we get this much more interpretable picture or map of how all the microbiomes are related. So let me explain what this map is. So each point on it is a location of a microbiome read out from its DNA and distilled just down to one point. And 2 points are close together if they're more similar in their microbial communities by UniFrac, that evolutionary distance metric, and they're further apart if they're more dissimilar in terms of their microbial communities, again measured by UniFrac. And we use an algorithm called principal coordinates analysis to take all of those distances and turn it into a layout in typically dozens of dimensions, but we usually show you the first 3 for simplicity. And remember, these are all healthy people, so there's no disease on this map. But what I can do is I can show you the main thing that's going on in this data set. And what you can see immediately is that the different parts of the body emerge almost like different continents on this map as being very different from one another. So to highlight this, if I show you the mouth and the gut of the first person in the HMP, you can see they're in completely different locations. And part of the reason why this was important is prior to the HMP, different researchers had looked at different subjects with different methods in different parts of the body, so we couldn't tell which differences were due to the method, which were due to the body site, and which were due to the different subject populations. But in the HMP, we used consistent methods for everything. So this tells us that the mouth and the gut are really different from each other, but you probably didn't need to spend $173 million to figure that out, right? But what we didn't know was the scale of those differences. And it wasn't until we did the Earth Microbiome project that we really started to figure that out, because what we could do is we crowdsourced tens of thousands of samples from across the research community. We asked what two samples on the planet's surface are just as different from one another as the mouth and the gut of this one individual. And the results were astonishing. So if you think of your if you think of your mouth as being kind of like a coral reef with complex mineralized structures covered with biofilms that maybe your dentist bugs you about from time to time, amazingly, your mouth is as far from your gut in its microbiome's ecology as the water in this reef is from the dirt in this prairie. That's amazing, right? Because it means a few feet along the length of your body is comparable in terms of the change in the microbial communities to thousands of miles across the Earth's surface, and we never expected that. Now, we can also look at journeys on this microbiome map. And so this leads to questions about where do our microbes come from in the first place? Where do we begin on this map? And if you have dogs or kids like I do, you likely have dark suspicions about that, all of which are true, by the way. So we can match you up to your dog with fairly high accuracy by looking at the microbes you share. But in all seriousness, how we're born has a tremendous impact on our first microbes. So in work we did with Maria Gloria Dominguez-Bello, was published in PNAS back in 2010, Gloria was able to sample mothers 20 minutes before they gave birth, and then their babies within an hour after birth. And what we saw is that if you're delivered vaginally, all of your microbes from all over your body look like your mother's vaginal community, whereas if you're delivered by C-section, instead, your microbes look completely different. They look like a skin community instead. And so you're essentially denied that microbial birthright with which our species has co-evolved for millions of years. And you might be wondering what happens after that. So in work we did with Ruth Lay, he's a director at the MPI now, we tracked the development of one microbiome of one child in the gut over 2.5 years of life. And so you can see that his fecal sample starts off in the vaginal region of the plot, which we would expect from how he was delivered. And what you're going to see is how his infant microbiome develops over the first 2.5 years of life as he starts to go towards the adult fecal community. And so the question is, how fast is this progression? How complete is it? How fast or slow? Is it smooth, or do things happen along the way? And I'll just start this going. So each frame in this is a week. You see sometimes there's just a little change one week to the next. Other times there's tremendous change. And remember that what matters most on this map is distance. And the difference between this kid's microbiome 1 week to the next can be larger than the distance between any two healthy adults in the Human Microbiome project. So if you think your kid looks like a different person 1 week to the next, that can be literally true in terms of his microbiome, which is where most of his genes are. And coming up here is something fascinating. So you see he gets antibiotics for an ear infection, and you see that radical change followed by recovery. And that went by pretty fast, so I'll just rewind it for you and play it for you again. On administration of oral amoxicillin, what you can see is this massive change in the microbiome undoing months of normal development. But in this particular case, the child is very resilient and within a few weeks bounces right back to where he was. So by the time he gets to 2.5 years, he's essentially in the healthy adult region of the plot. But that doesn't happen for every child or for every adult. And one thing we're very interested in at the moment is trying to understand why some people and their microbiomes are very resilient to antibiotics, whereas others are permanently altered after even a single course. And so understanding the impact of antibiotics and how they combine with our pre-existing microbiome and with our other medical conditions to have impacts that are more or less severe on different people, I think is tremendously important at the moment, although there's been relatively little research done on it at this point. Now, one thing that you can either see as a message of terror or as a message of hope is that the combination of antibiotics, C-section, and bottle feeding is particularly bad in terms of not just the microbiome, but in terms of phenotypes later in life, including adiposity at age 8, cognitive ability at age 12, and other things that happen throughout childhood, and some evidence suggests even throughout adulthood. However, the other way to read this is that breast milk in particular seems to erase a lot of these phenotype effects of antibiotics in early life and delivery mode. So the idea that we could have a nutritionally based solution to some of these problems, and that we might be able to identify whatever the active ingredients are in breast milk and be able to supply them in formula for families who, for whatever reason, are not able to breastfeed, that could be really important for limiting some of the deleterious effects of depleting our microbiomes early in life. And to do this at UC San Diego, we have this organization called MommyCore, led by Lars Bodie, who you can see a picture of there. And essentially what we're trying to do is we're trying to understand all the components of breast milk, including the beneficial bacteria, the human milk oligosaccharides, the antibodies, and so forth, to try to figure out how to confer those benefits for everybody. So I mentioned the Earth Microbiome Project very briefly before, and so what this was a large-scale effort to understand microbiomes from across the planet. And this was initiated by the US government at the DOE, which held a workshop to talk about it. But then actually doing it, it was mostly picked up by private foundations and in-kind donations from companies, for which we're very grateful. And one thing that was a big problem was that the technology to do something on the scale of 27,000 samples didn't exist when we started the project back in 2010. And so we had to develop a lot of standards, a lot of protocols, even new methods for storing all of the information on disk in what's called a sparse format instead of a dense format, using a tool called HDF5, which was developed by NASA to store massive data sets. And the result of all this, in Kathy's paper looking at the different kinds of environments that I showed you, that was back in 2007, and her study looked at 202 samples and about 21,000 sequences. But just a decade later, when we published the first paper on the Earth Microbiome Project, we looked at 27,000 samples and 2.2 billion sequences. And perhaps what's most astonishing is that today, when we run the NovaSeq instrument, so the latest sequencing instrument that we have at UC San Diego, every time we run that, which takes about 2 days, we get another 10 to 20 billion sequences. So 5 times the entire amount of the study that we published in 2017, that was at the time by far the largest study of its kind. So one thing we were able to do with this massive data set is we were able to develop an ontology where instead of imposing our preconceived notions about what wasn't important in the microbial world, we could use the similarities and differences among communities to tell us what was important. So like the free-living host-associated split, the difference between saline and non-saline samples, between animal and plant samples, and so on, turned out to be incredibly important for structuring these microbiomes around the world and with respect to each other. And this figure is pretty complicated. The only reason I'm showing it is to demonstrate that we recaptured all of the main features from that study we did back in 2007 using different studies and different techniques aggregated from the public databases. Whereas here, we're doing everything with the same techniques, and we recapture all those things like the saline, non-saline split, the host-associated free-living split, the animal-plant split, and so on, very clearly and overwhelmingly with tens of thousands of samples. But all of this has been based on DNA, and we really need to go beyond DNA and look at things like RNA, look at things like the metabolome, which tells us about all the metabolic capabilities and small molecules that particular microbes are producing, because that's what really gets us to function, rather than just getting a catalog of the name tags of the microbes that are in a particular location. And Jack Gilbert and Peter Larson, when they were at Argonne National Labs, we recently recruited Jack here to UC San Diego, where he's one of the first faculty members with a split appointment between pediatrics and the Scripps Institution of Oceanography, which is an unusual combination, but already a very effective one. And so what Jack and Peter were doing was using artificial neural networks to take environmental parameters and then model the kinds of bacteria that would live in each of those different environments. So this is a picture of the Western English Channel. So at the English Channel. At the top, you can see England. At the bottom, you can see France. And what you're looking at is you're looking at microbiomes of the ocean extrapolated over a whole lot of pixels and a whole lot of time points using ground truth measures from a couple of dozen samples. And so what's exciting about this is that even if you could, even at 75 cents a sample, you can't afford to sequence every patch on the ocean all the time, right? Just collecting the samples would be cost prohibitive. And so if we can link this kind of thing to satellite data and predict the microbiology, we can do everything at a much larger scale. So the idea is that we can take satellite modeling and modeling data, predict the microbes from them, then predict the relative metabolic turnover, and then look at feedback cycles for how microbial metabolism in turn affects the communities. And then applying this to CO2 turnover specifically, the agreement we got with the UNESCO Surface CO2 Atlas was excellent. We can also do this sort of thing in soils. So tallgrass prairie used to cover most of the central United States before agriculture, and it's essentially an extinct ecosystem. But Noah Ferrer at Boulder was able to find a few relics of it in graveyards and between railroad ties and look at the soil microbiology. And then Josh Ladow, who's at the Gladstone Institute, was able to do what's called niche modeling, so looking at historical climate records, including rainfall and temperature and so on, and predict what those microbiomes would have looked like throughout the entire continental US over the historical range of the tallgrass prairies. And so what's exciting about this is that even for an essentially extinct microbiome, we can reconstruct it and understand how it would have worked, and then from those relics, think about reseeding it across the country. We can also do this sort of thing on a much larger scale. So the Earth Soil System model is a community-produced model by hundreds of researchers where it predicts 12 parameters across the entire Earth's surface at relatively high resolution. And you can extrapolate it thousands of years into the future or thousands of years into the past. So look at the impacts of different climate and land use scenarios. And so what I'm showing you here is soil moisture. What we can do is we can predict from that set of 12 parameters what the microbiomes would look like at each patch in the soil. So here I'm showing you the alpha proteobacteria that like it dry. So you'll see them in the Atacama, you'll see them in the Sahara, then the Gobi, then all over Australia. We can predict from who's there what the functions are using an algorithm called PyCrust that we published in Nature Methods back in 2013. And then we can take, so here I'm showing you an enzyme that degrades cellulose. And then we can predict the relative metabolic turnover at each site. So here I'm showing you CO2, but I could show you any one of over 2,000 metabolites. And so this modeling approach is very useful for understanding what's going to happen to the planet at a microbial level under different climate and land use scenarios. We can apply these same sorts of technologies at the scale of our own body. So here's a photo of Peter Dorrestein, who I mentioned earlier. He's my primary collaborator on mass spec, and his postdoc, Mina Boslamini. And Peter's been very interested in the skin as an interface between ourselves and our environments. And he was able to recruit one male and one female subject who were willing to sample themselves at 500 locations over the body. And then Peter ran it through two kinds of mass spec to look at the metabolites. My lab ran DNA sequencing to look at the bacteria. So when we do this over the body, again, the scale is from blue as the least to red as the most. These are unidentified mass peaks, so we don't know what these metabolites are, but you can see some of them are broadly distributed over the body, others localized to the armpit, the groin, the toenails, or whatever. We can also do the same thing with the microbes, and this is exactly what you'd expect from classical microbiology. So staph likes it moist, and we see it in the nostrils and on the feet. Propionibacterium is more on the head and shoulders. Corynebacterium is more broadly distributed over the body. You'll notice that only half of the body is displayed for the male and for the female. Their original plan was to do 1,000 sites all over the body, but they started early in the morning. They were still at it at 8 o'clock in the evening. They realized they were going to be up all night if they did the other half of the body. So they just collected half of it, but the results are nonetheless very interesting, although we can't look at things like symmetry, unfortunately. Anyway, what we can do is we can start to use the spatial co-localization to figure out which microbes are doing what in the environment. So I told you Propionibacterium was on the head and shoulders. Oleic acid is there, but it's also on the hands. But then it only oxidizes on the head where there's Propionibacterium, and not on the hands where there isn't Propionibacterium. We see a similar pattern with palmitic acid. So what we can do is we can conjecture that the microbe and the breakdown product that are in the same place mean that the microbe is catalyzing that reaction, and test that in the lab. And so when Peter did that using reference standards for the molecules, you can see oleic acid is only produced when you incubate the bacterium that we think makes it and the chemical precursor. And then it also oxidizes only under those conditions. And so what's exciting about this is that we can develop this into a high-throughput technique where anywhere that we can get this kind of spatial data, we can start to understand what microbes are doing what chemical reactions, and what the functional implications are of having those microbes in a given location on the human body or on the planet. More recently, we've been able to take this inside the body in collaboration with Doug Conrad's lab, as well as a number of others. And so, what we're looking at here, and this just shows you the sheer range of disciplines that are represented by this collaborative project. What we're looking at here is what chemical environments do microbes in cystic fibrosis reside in inside the lung. And you might be thinking, wait, isn't cystic fibrosis a classic example of a genetic disease? And sure, nothing's changed about the genetics of cystic fibrosis, but the difference between living to age 2, which was typical a few decades ago, versus living to age 42, which is the average age of Doug Conrad's patients now, is almost entirely in better approaches to managing the lung microbiome and managing the polymicrobial infections that kill CF patients. So a very important problem. A lot of these pathogens have been studied very extensively in the lab, but what we wanted to know is do they actually do the same thing in situ in the lung as they do in a petri dish, or do they do things that are completely different? So to address that problem, we took 4 of Doug's patients who were undergoing transplant surgery. And so we took the CT scan of each patient and then the explant lungs that we could dice up into little cubes and run each cube through sequencing and through mass spec so we could figure out what the microbes were and so we could figure out what the molecules were throughout the whole lung that was being removed. And Theodor Alexandrov, who was then in Peter's lab but is now at EMBL, developed the software called ILI, which basically allows us to take the CT scan, dial up and down the transparency, and then look at any microbe or any metabolite that we're able to measure throughout the 3D structure of the object we're looking at. In this case, the lung, but you can apply it to any object in 3D. So one thing we found is that the classic Pseudomonas quinolone signal that it uses for quorum sensing, we did not find that in any of these lung samples. What we instead found was some other quinolones, NHQ and NQNO, that are not considered the canonical signaling molecule and are related to PQS. We can find that by this technique called molecular networking. And so what we think is that there might be completely different quorum sensing modalities going on in the lab versus in the lung. We can also start to relate these things to each other. So if I show you Pseudomonas and then 2 other bacteria, Granular Catella and Staphylococcus, you can see that this new molecule, so you can see Granular Catella is high where Pseudomonas is present but relatively low. And similarly, Staphylococcus is high where Pseudomonas is relatively low. And that signaling molecule, NHQ, where it's produced, and we're pretty sure it's being produced by the Pseudomonas, where it's being produced is not where the Pseudomonas is highest, but rather where it's battling it out either with Granular Catella or with Staph. So we think it might be involved in that microbe-microbe interaction rather than in quorum sensing within Pseudomonas. And that's something we're following up in the lab now. We can also see pharmaceutical degradation. And so if you take something like piperacillin, how it's broken down is completely different in different people's lungs. And we think that, again, that's mediated by the different kinds of microbiomes that those lungs have. And in general, a huge number of drugs have just in the last couple of years been shown to be modified by the microbiome and modified in different ways by different microbes in the gut. So this is probably true throughout the rest of the body as well, and is a very important emerging area to watch where we might get a much better understanding of which drug you should prescribe for which patient based on how it will interact with the microbiome, which in turn is driven in large part by the diet and other lifestyle factors that you and they can potentially take control over. Let's see, we also see the same antibiotic penetrating very differently into different people's lungs. We also see penetration of environmental chemicals like phthalates, for example, and phthalates attach to different size particles depending on which phthalate it is. So this gives us an estimate of lung function by being able to see how far different sizes of particles are able to penetrate into the lung. Now, at the moment, all of this, remember that we have to dice the lung up into little pieces in order to be able to do this. So we can only do it once and only for a lung that's not being used anymore, right? But what we're hoping is that we can use some of the same modeling techniques that we use in the Earth Microbiome Project, where we can take non-destructive sampling, like from sputum, for example, or maybe something a little more invasive like BALs, and then build a model of what's going on microbiologically and chemically inside the lung in 3D that we can then use for diagnosis at a much earlier stage rather than only once the lung has been removed. So coming back to the scale of our planet, one thing that's amazing is how atmospheric aerosols connect even different continents to one another. And this is satellite imagery from NASA showing dust flows across continents. And the timescale of this is that it takes maybe a couple of weeks for the dust to get across the Atlantic from Africa to North America, for instance. But that dust carries a tremendous amount of stuff with it, including live viruses, live bacteria, pollen, and all kinds of other substances. So Kim Prather, who directs a centre called CASE, which is an atmospheric aerosols centre supported by NSF, and I have started collaborating recently looking at what's in these aerosols. And so Kim had been doing this project called Cal Water, where essentially she was flying planes through clouds to collect the aerosols and understand how they affect precipitation in California. And back in 2013, she built on some work that Noah Ferrer and Bob Biles and I had done back in 2010 and 2011, looking at ice nucleating microbes in clouds. And she was able to find these atmospheric rivers that connect Africa and California. And what she found was that amazingly, the snowpack on the Sierra Nevada range is largely driven by particular microbes that come over from Africa across the Pacific and nucleate ice crystal formation. So our climate can be driven by processes that are happening on another continent. Now, climate isn't the only thing that these atmospheric microbes are able to modify. So this is Jane Burns, who runs our Kawasaki Disease Research Center here at UC San Diego. Kawasaki disease is the most common form of acquired heart disease in children. And what you can see here is in Japan and in San Diego and in Hawaii, Kawasaki disease incidence in red, and then the amount of wind that's coming from the northwest direction in blue. And this trend is so strong that Jane actually knows when she needs to come into the clinic based on the weather report, she says. And what we think is happening is that there's a source in northeastern China of some etiological agent, possibly a fungus or possibly a bacterium, that's triggering Kawasaki disease exacerbation or possibly causing Kawasaki disease in the first place. So together with Kim and Jane, at the moment, what we're doing is we're trying to match the microbes that we isolate from clouds and from aerosols up to the microbes under the fingertips of children who have this characteristic fingertip peeling with KD, as well as microbes in their airways, to try to understand what the relationships are between the environmental microbes and the microbes that may be causing this health impact. We're also, I don't want to scare you about exposure to microbes in the outdoors though, so another project we've been doing with Dorothy Sears and a number of others is this project called Nucleotides to Neighborhoods, where we're trying to integrate everything from DNA sequencing up to things like walkability and food deserts at the neighborhood scale to understand what has a big impact and what has a small impact on health and gene expression. And so working with the American Gut Project, Dorothy was able to show with these very detailed traces of where each subject went, that even things like the number of minutes you spend outdoors has a substantial impact on your gut microbiome, where the more minutes you spend outdoors, the more diverse the microbes are in your gut. And in general, microbial diversity in your gut is a good thing associated with good health states. Now, when we think about sources of microbes in the air, it's very easy to think about terrestrial dust just because it's so visible. But what covers way more of our planet's surface is water, and hardly anyone has even thought about how marine aerosols might contribute to bacteria in the atmosphere that we're breathing in, or that may be modifying our climate. So one thing that Kim built is this wave flume, which is up at the Scripps Institution of Oceanography, which is part of UCSD, where what it does is it's able to recapture breaking waves in the lab. What's amazing about this is if you just get a jar of seawater and you shake it up, it's random which bacteria and viruses are in the aerosol and which are in the bog phase. But if you break the waves with the right power spectrum and you get the particle distribution of the aerosols right, it's extremely non-random which viruses and which bacteria are able to launch themselves into the sky. So we're analyzing those at the moment, essentially looking at what microbes transfer from bulk seawater to the sea surface microlayer, then ultimately into the sea spray aerosols, and then trying to understand which of them have good and which have bad health impacts and climate impacts. One thing we've been doing, and this was published in Nature Communications last year, What we're doing here is just looking at the different kinds of microbes to look at which are enhanced in the aerosol and which are diminished in the aerosol based on their phylogeny. And what's interesting about this, if I just zoom in on a piece of it, is that actinobacteria like Corynebacterium have very waxy coatings that make them especially hydrophobic and make them able to get into the aerosol phase very effectively. And so what we're doing is, at the moment, is we're screening these and other bacteria for ice nucleation properties. So there's a lot of implications connecting oceans and human health, ranging from things like toxic compounds that accumulate in fish fat. On a project I won't talk about, where we're collaborating with Merete Expo at the Norwegian Institute for Public Health, looking at breast milk in Norway. The breast milk samples that we're getting have so much mercury, so much cadmium, and so many PCBs in them that we have to incinerate them as toxic waste after we're done looking at them when they come into our lab. And it's terrifying to think that that's what infants are feeding on. So things like that, things like biosynthesis of different compounds in the ocean, things like sewage-contaminated water and sewage-contaminated aerosols are becoming more and more important as factors. And so one thing we're working on at the moment is following up on some of Kim's dye tracing experiments in the Tijuana River, where basically the question is how far does that dye come inland just with the winds, and also are microbes coming in on that, and how does it interact with other stuff like ozone. And so what we're doing is basically going everywhere from the beach to way inland and doing sampling to see if we can detect different chemicals that are coming in and also different kinds of microbes that might be coming through in these sewage events. So the overall vision here, and what's really going to connect environmental and human microbiology on an even larger scale, is continuing to decrease the cost and making it more accessible. And I'm really inspired by some other technological examples in this respect. And for example, Tanya Yatsenenko, one of our collaborators on a Gates-funded malnutrition project with Jeff Gordon, took this photo in Malawi back in 2009 at the malnutrition clinic that we work with. It's run by Mark Minari. And even in 2009, although this was a population where food was very scarce, you see that they did already have access to cell phones. And what's amazing about this is that there are over 9 billion active cell phones on Earth, which is pretty impressive when you consider that there's only about 7 billion people. And of the billion poorest people in the world, more than 20% of them have their own cell phone. And this is true everywhere, not just in Malawi. So I took this photo in Bangladesh in 2012 when I was visiting ICDDRB, one of the world's best digestive disease research centers. And you can buy a sizable hunk of cow, and then right next to that you can get your cell minutes recharged, which is what that orange sign is, what that yellow sign is. And perhaps most remarkably, when I was in 2014 working with a Hadza, I had the remarkable privilege of shaking hands with a man who just a couple of weeks before had shot a giraffe and traded its skin and some of its meat to members of the Datoga, the next group over, for the cell phone that you see him holding there, where he keeps the minutes and the electricity topped up by trading wild honey that he gathers. So the Hadza hunt and gather 95% of their calories, and he gathers wild honey and trades it at the trading post for electricity in minutes. And so this is a technology that's penetrated everywhere on Earth, including some of the most remote regions. And a large part of the reason why is Moore's law. It's just got cheaper and cheaper to do the signal processing that ties together a cell network, and it's become more and more robust and deployable. But one thing that's getting cheaper even faster than computation is DNA sequencing. And this graph from the NIH just shows that in the period that computation has got 100-fold cheaper, so this is on a log scale, DNA sequencing has got more than 10,000-fold cheaper. And if those trends continue, we could imagine that maybe DNA sequencing or mass spectrometry is going to be as robust as the technologies that I'm sure each of you are carrying around today, where the CMOS sensor that drives the camera in your cell phone, just 20 years ago, that would have been at the heart of a classified multi-billion-dollar spy satellite, whereas now you can just use it to take video of your grandkids or snap a photo of your shopping list or whatever. And if we can do that with these molecular readout technologies, the way it's already been done for light readout technologies, I think that will really transform society and our relationship to our health and to the microbial world. So there's a lot of organizations to thank here. I'm just going to show this slide briefly rather than reading them all out. Also the Center for Microbiome Innovation that I direct, that now has over 140 faculty associated with it, and a dozen corporate sponsors, including Metagenics, which we've been very excited to work with. And the work here represents literally over 1,000 collaborators, who again I won't read out a long list of, but I mentioned many of them during the talk. It's also a true pleasure to work with the numerous people in my lab who are incredibly talented and hardworking, and our many sources of funding, including tens of thousands of members who've signed up for the American Gut Project, which is still very much collecting samples. And with that, I think we're going to save questions for the fireside chat. Is that right? Yep. So think about your questions. We'll talk about them in the fireside chat. Thanks again for coming along this morning, and thanks again for inviting me back.